Hi, everybody. I think we're about ready to get started, so I'm just going to take a brief moment to go over what we're going to be covering today. My name is Barbara Hill, and I'm going to be your host today. Leading today's webinar is going to be Thorin Tabor, who is the lead developer for GenePattern notebooks. In our webinar, we're going to provide an overview of the HCA Notebook Workspace. Then we're going to take a deeper dive into a few of our featured notebooks. Then we'll review some additional features and future plans. And as we're progressing throughout the webinar, feel free to ask any questions anytime via chat. You can either ask them privately to me or you can ask them to everybody in the group. We're going to try and answer those questions as we go along, but if we have good momentum, we might just leave them to the end, but we will definitely answer all of your questions. To that end, there will be a Q&A section at the end of the webinar. Also, some people have asked this already, but there will be a video of today's webinar made available to everyone, and that will be a link will be sent out with that once the video has processed. So I think that's all that I need to cover. So I'm going to hand it over to Thorin now.
Hello, my name is Thorin Tabor. I'm the lead developer of the HCA Notebook Workspace, which is an integration of Jupyter Notebook environment and the GenePattern platform for integrative genomics. It also includes special features for accessing the growing repository of single-cell data hosted by the Human Cell Atlas's data coordination platform. In the webinar today, I'm gonna briefly go over the platforms that we integrate, Jupyter and GenePattern. I'm also going to talk a bit about how they can be used to access HCA data. And then I'm going to walk you through several bioinformatic notebooks along the way, pointing out the features that the Notebook Workspace offers and how you can use those features in your own research. So I'm going to start off by talking about Jupyter. Jupyter is a popular open source framework for scientific computing. It enables interactive computation in collaboration by encapsulating code, programmatic output, and rich text annotations in a notebook document. Essentially, this means that you can run through your analysis, check or plot your results, and explain your methodology all from the same document interface. And what's more, the HCA Notebook Workspace makes it easy to share this document with your collaborators or to make it publicly available to accompany a publication. Finally, Jupyter has a rich ecosystem of features integrations, such as the ability to embed images in interactive widgets, and the ability to call the command line or other programmatic languages from a notebook. And these I will go over in more detail later. The other platform the Notebook Workspace integrates is GenePattern, which is a platform for reproducible bioinformatic research. It's a long-established project. The first release was back in 2004, and it's been going strong ever since. Like Jupyter, it is open source and has proven to be quite popular. One of the key features of GenePattern is it provides hundreds of analytic modules. I think about 250-ish at last count. It bundles these analyses up, including all of their dependencies and environment, and it lets you run them on a dedicated compute cluster. What's more, all of GenePattern's analyses can be launched from a familiar web form sort of interface, no programming required. Analyses it provides include the most of the popular methods in genetics, genomics, transcriptomics, and proteomics, as well as general machine learning methods. It's got some 60,000 registered users, and the GenePattern public server usually runs somewhere in the ballpark of 4,000 analyses each week. Finally, there's a repository of community-contributed methods that cover a wide range of techniques, including CRISPR analysis, bisulfate sequencing, flow cytometry, and RNAi screens. So what we've done is we've taken all these tools, Jupyter, GenePattern, and the HCA's data coordination platform. We've combined them together to get the HCA Notebook Workspace. We want to make it easy to pull data from the DCP, to run it through your custom analysis or one of the ready-made analyses provided by GenePattern, and we want to make it easy to share your results and methodology with others, either privately with your own collaborators or publicly to accompany a publication. One of the things that we are doing to support this is that we're building a library of single-cell notebooks that will encapsulate all the facets of single-cell analysis, from pre-processing to clustering, and from visualization to trajectory analysis. Many of these approaches developed in coordination with the HCA. Our goal is to provide all of the tools necessary to encapsulate a complete research narrative, from its conception to its dissemination. This includes the ability to integrate and install programmatic libraries, the ability to embed tools such as GenePattern, IGV.js, or Cytoscape.js, and the ability to interleave text, visualizations, interactive widgets, and graphics along with the analytic content. So now you have the high-level view. Rather than continuing to simply talk about it, I'm going to jump in and show you the Notebook Workspace hands-on. So the first thing I would do is go to hca.genepattern.org and log in. I've already done that because I wanted to get everything pulled up and ready for the webinar. But it's simple to either sign in or register for a new account. And let me switch to the browser here. Oops. There we go. Once you're signed in, it should bring you to your private notebook workspace. Think of this as a folder for storing your notebooks and associated data. Here you can create a new notebook, open an existing notebook, launch a terminal, create or edit a text file, etc. As a brand new user, it should come with a couple tutorial notebooks, but in my case, there are a number of other notebooks that I've been working on. To open a notebook, just click on its name in the list. To create a new notebook, click the New button, and then select the Notebook option. You'll notice that there are a number of tabs at the top of the page here. One of these tabs is called Notebook Library. This tab contains a variety of public notebooks that have been made available on the Notebook Workspace. This includes all of the Human Cell Atlas's vignettes, which are short scenarios for working with DCP data. Also included are all of the notebooks that I will be demoing today. To run a copy of a public notebook, all you need to do is find the notebook you want to run, either by browsing the list or using the search box in the upper right, and then you click on an entry and select Run Notebook in the resulting dialog. From this tab, you can also access notebooks that have been shared with you by your collaborators. Just go down to Shared with Me and click on it and view the notebooks shared with you in the list. Your list will be empty, but here you can see that 2 notebooks have been shared with me. To run one, all you need to do is click on one of those notebooks in the list. Once you run a notebook this way, it will appear in your private workspace, and you can run it from there in the future. You could also take a notebook that you've developed and share it publicly with everyone to accompany a publication or just because you want to make it freely available. To do this, click on a notebook that you have there in your Files tab and click the Publish button. This will pop up a dialog where you just fill out this simple web form, giving it a description, an author's name, a quality, etc., possibly some tags, those are optional. And then you click Publish. And when you click Publish, it will make that notebook available in the notebook library that you saw in the last tab. So the first notebook that I'm going to show you is a demonstration of the DCP expression matrix service. This notebook allows you to search the DCP for datasets based on species and tissue. Once you've found a dataset you want, you can then send it to the expression matrix service, and it returns an expression matrix in a format of your choice. So let's take a look. The first thing you'll notice when viewing this notebook is that there are some instructions at the top, and there is this interactive widget. These instructions explain the purpose of the notebook and how to perform the first step when searching the DCP. Now, all notebooks are made up of cells, and this is called a markdown cell. If you double-click it, you can see the source of the cell, which can be either HTML or Markdown. One of the goals of the Notebook Workspace is to make it easier to collaborate with researchers of all levels of technical sophistication, from machine learning wizards to bench biologists. One of the features we've added is the ability to edit these cells with a familiar what you see is what you get rich text editor. To access this feature, just click the Rich Text Editing button on the side. Now, you can see the cell displayed inside this editing frame, and we can now edit the content much like we would content in Microsoft Word. We can make things bold, we can add new paragraph styles, we can add links, etc. Once you're done, just click the Run Cell button on the side, and we can leave editing mode entirely. All right, so enough of the instructions. I'm going to try searching the DCP. In this case, I want to look for human pancreatic data. So let me select Homo sapiens and pancreas from the list here. And then I just click the Run button. So, oh yeah, a technical issue to note. If you visited the Human Cell Atlas's data portal this morning, you'll notice there's this big yellow banner across the top of the page warning you that the DCP is in maintenance mode right now for data re-ingestion. So it looks like I've returned a couple results and that's sufficient here, but if we run into any issues with this particular notebook, it's because we're querying the Data Coordination Platform's API and it's in maintenance mode today. All right, so it looks like we've got some search results. So what I want to do is I want to scroll down here to this next step. And here's some more instructions in the GenePattern widget. In this step, I'm going to select the data I want to send to the matrix service and the format I want it in. So you'll notice that when I click the first parameter, it automatically populates with all of the compatible search options. So I select the one I want, and in this case, this one right here, and then say I want it in MTX format. I then click Run. This will kick off a matrix service job doing all the processing required. And you can see that this job is represented by this new widget. As the matrix service job runs, this widget will query its status every 30 seconds or so. And once the file is ready, it will display a link below. For this data set, this will take somewhere in the 1 to 3 minute range. So in the meantime, let's take a closer look at this widget. All of the widgets you've seen so far have been generated by GenePattern's UI builder. Essentially, this is a way to turn any Python function into an interactive web form that can be launched with the click of a button. So let's take a look at the code we're running. We can do this by going to the Gear menu and selecting Toggle Code View. So here's the actual Python method that's being called when the Run button is clicked. As you can see, the parameters in the form correspond to the parameters of the function. All that we need to do to turn the function into an interactive widget is to add this decorator right here, notebooktools.buildUI. From that, the entire widget is automatically generated. And then this data structure right here is optional. But you can use it to override the display name of the parameters, give them well-formatted descriptions, specify a drop-down list of options, etc. And you can see how we're specifying our list of choices right here. The UI builder has a lot of features and options. I could probably run a whole workshop on it alone. But regardless, all of its documentation is up on the website. Right now, the UI builder is Python specific, but we have an R version that is not quite released yet. But it's in the testing phase and should be out in the near future. All right, so I've just toggled the code view closed again, and it looks like the matrix service job is still running. It's probably got another minute or so until it completes. But rather than sit and wait for it, I'm going to move on to the next notebook. Either way, once you have the matrix file, it's easy to save it or send it to a downstream step of your workflow, such as pre-processing or visualization. The next notebook I'm going to be demonstrating is a workflow that clusters single-cell RNA-seq data. The purpose of this notebook is to provide a standard single-cell RNA-seq workflow for pre-processing, identifying subpopulations of cells by clustering, and exploring biomarkers to explain intrapopulation heterogeneity. It's modeled after the Seurat guided clustering tutorial. and performs analyses using the ScanPy library. In this case, I'm going to be using a data set of about 3,000 peripheral blood mononuclear cells. However, I'm not going to delve too much into the biology and machine learning contained in this notebook, as the primary purpose of this webinar is to demonstrate the tools that were used to create it and which you might use in your own research. So if you're interested more in the science, the article to this notebook is linked right here at the top. And the first thing I want to note about this notebook is that it's significantly longer than the last one. In fact, one of the features the Notebook Workspace adds is a table of contents. And we can pull that up by clicking on the Table of Contents button right here in the toolbar. You can see this opens up a side panel. And in the side panel, we can see all of the steps in the notebook. And you can even scroll to individual steps by clicking on them right there in the Table of Contents. If I want to add an inline table of contents, this can be generated by clicking on the gear button right there and selecting the Add Notebook Table of Contents cell. And it will automatically generate a table of contents and add it as a cell to your notebook. So, as I scroll through this notebook, you can see that there's a diagram of the workflow it uses. It's easy to add images like this to notebook documents. Just drag and drop them onto the editor, and it will automatically upload and embed them in your document. All right, so scrolling down, you can see that here is another UI Builder cell. These should begin to be familiar to you. This is the third time we've seen them. In this case, when I click the Run button, it will download the data in MTX format and prepare it for downstream analysis. This step is also going to run some quality control metrics. And now you can, oh, it's running. Give it a second here. And now you can see it's returned an interactive set of widgets here that allow us to filter by the number of genes, the total counts, and the percent of mitochondrial genes. And we can do that simply by changing these range sliders and giving it a few seconds to make adjustments. Okay, in this next step, I'm going to do some pre-processing. This will do some quality control and log normalize the data, as some of the downstream steps assume a normal distribution. It's also going to perform PCA, which is a dimension reduction algorithm. It's going to allow us to pick out a few principal components that explain the bulk of the variance in the data set. Here you can see the PCA, plot. And as you can see, the 5 or so principal components here explain the vast majority of the variance in the data set. So this next step, I'm just going to continue on here, takes about a minute and a half to process. So I'm going to hit Run first. This step is going to cluster the cells using the Louvain method for community detection. It's then going to produce a t-SNE visualization so that we can see the clusters. In this case, we're only using the top 10 principal components, but we could have filled out the form differently and used a different number. Give me just a second for it to run here. All right. You will notice that this visualization is interactive. As we move the mouse over the data points, it gives us their graph coordinates. We could also rerun this step using a different resolution, complexity, or number of principal components up above. In the interest of time, I'm going to run through step 4 somewhat quickly. But what the notebook is doing is it's exploring genes that are differentially expressed in clusters as potential biomarkers. In doing that, it's producing some plots to explore the data. In this case, we have a heat map. It's going to reduce more t-SNE and violin plot. And then it's going to compare the expression between clusters with a volcano plot. So again, if you're interested in what the sciences is doing, then the article is linked from the top. But you can see it's easy to produce these sort of interactive visualizations and embed them in a notebook document. Finally, the notebook lets you serialize the data to disk so that you can use it in other workflows. That's done right here in step 5. And it's done. It's exported to our data analysis folder. For example, if you went there to your workspace, you can see that right there, there's the data folder that it has put your results in. The third notebook I'm going to show you demonstrates how to perform hierarchical clustering on RNA-seq data, along with a pre-processing step and visualization. In this notebook, instead of performing the compute for these analyses in a notebook kernel, we are going to send the analysis to GenePattern's public server's compute cluster and then retrieve the results. That is, this notebook runs hierarchical clustering as a GenePattern module. This workflow allows us to run computationally intensive analyses in an environment that meets the needs of the algorithm. In this case, we're sending the analysis to a compute cluster in the Amazon cloud. But GenePattern also has a high-throughput compute cluster hosted by Indiana University. As you look at this notebook, the first thing you may notice is this blue bar. This is a GenePattern authentication widget. It's collapsed right now. Well, interesting. I will debug that in a second. It's collapsed right now, which is why it appears as a bar, but I can expand it to show the GenePattern system message. But essentially, this widget means that we are currently authenticated with the GenePattern server. You can see my username here, and the URL of the GenePattern server right over here. Scrolling down, here are the links to the data files that we'll be using in the notebook. And below that is our first analysis step. I'm going to run a GenePattern module called voom normalize. To do that, I'm going to drag and drop the links from our data to the appropriate inputs. And then I'm going to click Run. As you can see, this widget represents a GenePattern provided analysis, and it behaves more or less like the UI Builder widgets you've already seen. Only in this case, the widget has this blue color, which is a bit of color coding to let you know that the analysis is running remotely on the GenePattern server. This particular step is doing some pre-processing to fit the data to an approximation of a normal distribution, which is what the downstream steps expect. You'll notice that when we started this analysis, the widget collapsed and then a new one appeared. This represents the state of the analysis on the GenePattern server. It waits for a short time in the job queue before running, and eventually will display the results of the analysis. So this will probably take a minute or two to run, and then let's give it just a second. In the meantime, once it runs, I can move on to step 2, and I'm going to send the output of this particular step to the downstream step. There, it's finished running, and I've got that little notification. So moving on to step 2, now that the data is normalized, I'm going to send another analysis to GenePattern, this time doing the actual clustering. To do that, I click on the input parameter here, and you'll see that it automatically populates with the possible inputs. Alternatively, I could click on the output file, and it will pop up a menu. And I can select to send to an existing GenePattern cell or a new GenePattern cell. Or I could drag and drop and bring it down. Recall, all of these options are send to and come from functionality. It makes it easy to string analyses together into a multi-step workflow. Now, I would click the run button to start the analysis, but this particular step takes about an hour to run, and we don't have time for that in the webinar. So instead, I'm going to do the cooking show thing, where you put the you put the dish in the oven and then you cut to where you take it out again. So I already ran this analysis before the webinar. And if I collapse the widget here, you can see the results below. The results are a number of different files, which is basically with the data from hierarchical clustering. And if we want to see the data we put into it, that's easy to do. You can click on an output file into that menu, just like before. One of the options is Send to Data Frame. If we just click on the file and click Send to Data Frame, what it's going to do is it will generate a bit of code which loads the result file into a pandas data frame and then displays it in the notebook. The table is a bit big for the screen, but we could do that right here, and you would see a table with the matrix of results pop up. And I want to note this works seamlessly with any Python code that uses a pandas data frame. All right, so now before I get to the conclusion, I want to reiterate that the HCA Notebook Workspace you saw today runs in the cloud. We provide hosting and compute for your analyses, and you don't need to install anything. Just visit hca.genepattern.org and log in. However, if you don't want to run things in the cloud, you don't have to. This entire service is open source and freely available. You can install all of our tools you saw today on your local computer through either the pip or conda package managers. We also have the full stack available in a Docker image, and that's up on Docker Hub. You could run a notebook for just yourself, or even set up your own instance of the Notebook Workspace for your lab. And finally, the source code for all of this is on GitHub. Documentation is available right on GitHub and linked from each repository's README. I also want to note that the HCA Notebook Workspace and its associated frameworks have more features than I had time to demonstrate in this webinar. The workspace comes with a variety of useful programmatic libraries already installed. These include the HCA client, BioPython, Pandas, Plotly, scikit-learn, the interactive IGV and Cytoscape widgets, libraries for managing analysis jobs and working with common bioinformatic file types. It also supports R through the use of R cell magics. Compatibility with JupyterLab, which is the latest advanced Jupyter interface, is in the works. It will include real-time collaboration functionality. Through GenePattern, the workspace also supports a variety of interactive visualizations for hierarchical clustering, feature prediction, heat maps, and more. So before we move on to the Q&A, let me give a few acknowledgements to people who have contributed to the Notebook Workspace and the GenePattern. You can see the team on the left, Jill Meserov is our PI. On the right, you can see that we've gotten funding from these wonderful institutions. I would also like to put up a few resources from the net, which you can visit if you want more information. These include the Notebook Workspace, our website, our GitHub organization, Docker Hub, etc. Finally, I want to thank everyone for attending the talk. So with all that being said, I am happy to take questions.
So we have one question from Nikolai Thorin. He's asking if we have version control built into the notebooks. Do you want to take that?
Yeah, so we have version control in as much as Jupyter, the Jupyter framework supports version control. What you can do is from a notebook, you can set a checkpoint, and then you could flip back to that checkpoint. From the command line, which is accessible right from the notebook, you can also do any sort of git checkout or push or pull the different Git repositories. And that's another way to do version control if you're familiar with Git. Any other questions?
Well, if there aren't any other further questions for the webinar today, I think we will end that here. Feel free to reach out to us via the forum, which you can get to by going to genepattern.org/help, and that will answer any questions there. We will also be sending out an email with the video for this webinar for anyone who would like that once that has processed. So thanks to everybody for attending today, and we hope to see you again soon.
Thank you.